here. Uh, my research interest is on molecular mechanism of apical hook development. I'm working currently in National Institute of Plant Genome Research, New Delhi. So this work I did when I was postdoc in Professor Rishikesh Bhalerao Lab, Umia Plant Science Center, Sweden. And I'm still continuing this work in my lab. So as you heard about the differential growth in last 3 days, and this is 4th day, that there are 3 beautiful examples of differential growth: phototropism, gravitropism, and the third one is apical hook development. So differential growth means, so the one side cell elongate faster than the another side cell. So in this case, in the apical hook, the outer side cells, these cells elongate faster than the inner side cells. So how this differential growth takes place, this is a key question of my research interest. So what is the importance of this apical hook formation? So apical hook is formed when seeds germinate and young seedling emerge from the soil, and this protects shoot apical meristem from being damaged. So, so hook is formed in first 24 hours, then it is maintained another 72 hours, and then it slowly starts opening. I will show you a movie if it works. I think it will not work here, but so the angle which shown here in the blue color, we measured the angle, this angle, and when the hook is completely closed, this angle is 180 degree. So hook development is dependent on carrier-mediated asymmetric oxygen distribution. It is well known that oxygen form oxygen response maxima, and that it can be just by oxygen responsive synthetic promoter DR5. So oxygen is accumulated on the lower side of the hook, you can see here. And so this asymmetric distribution of oxygen regulate the hook formation. But how this asymmetric distribution is regulated through the polar oxygen transport, it is well known. And it has been also established well that this oxygen transport is through oxygen influx and efflux carriers. So oxygen influx are OX and LAX, and efflux are pins carriers. And when you see in the double mutant, then the hook development and is affected. So, and the several labs have outlined the hormonal pathway which is involved in hook development. But what is the downstream of these hormones? It is unknown. We believe that the downstream is cell wall, because it is for differential cell elongation, it is likely that cell wall modification is differentially regulated. So I will give you a very simple and probably simplistic view of cell wall. Cell wall is made up of cellulose, hemicellulose, and pectin, which involve RG1, RG2, and Hg. So these are the key components of biosynthetic pathway of gyroglucane, which involve XXT, MUR3, MUR2, and MUR1. So MUR2 is a fucosyl transferase, which is required for, which produce L-fucose, which is required for gyroglucane. But MUR12 is a GDPD mannose 4,6 dehydratase. So this L-fucose is required in pectin RG2 and gyroglucane both. So I will focus more on MUR12 in my presentation. So MUR2 is a dwarf plant and show bushy appearance. And interestingly, so this is L-fucose deficient mutant, but interestingly, when you provide boron, it rescues. But why in the presence of boron, it rescues? Because boron stiffens the cell wall and it makes RG2 dimerization. So first we check whether this defect due to the reduction in pectin RG2 dimerization, it lead to the defect in morphogenesis. And we've checked 2 alleles, and we found that in MUR1, both alleles show apical hook defective. And interestingly, when we provided the boron, it rescued it. So how boron rescue it? It need to be identified. And like because it was a fucose deficient mutant, so when we provided the boron, then of course it should be rescued, and it's rescued. And as I have shown earlier, that L-fucose is required in xyloglucane and pectin both. So we want to check whether this defect is due to xyloglucane or pectin. So mu2 provide the L-fucose in xyloglucane, and it doesn't show any defect. This suggests that the MUR1 hook defect is due to RG2 pectin. So as we know that differential cell elongation takes place outer side faster elongation and inner side slower elongation. So we checked the cell elongation rate in the MUR1 hook, and we found that in the wild type, you can see in the outer side and inner side. In MUR1, it is more or less outer side and inner side are same. And interestingly, when we provided the boron, it is like a wild type. So how boron rescued this defect? It is the question. So as I have shown earlier, that oxygen response maxima is necessary, and it so we checked DR5, and we found that DR5 signal intensity is reduced in MUR1, as you can see here, significantly it's reduced. And when you provide boron, it significantly goes like a wild type. But this oxygen response maxima is maintained by polar oxygen transport machinery, which involves pins and OX. So I checked the pin 3, pin 4, and OX plasma levels, and I found that all three decrease in MUR1, and in the presence of boron, they goes high. So why they decrease, what is the reason? Then we checked the transcript level, and we found that in the transcript level of these three decreased, and in the presence of boron, they goes up. We also checked other like LAX3, but they did, LAX3 did not change in MUR1 background. So most interestingly, like then because ARP7 is oxygen response factor 7 and ARP19 is oxygen response factor 19, both are crucial players for hook development. It is well established concept. So ARP7 and ARP19 also has hook defect. You can see here, it also has hook defect similar to MUR1. And then we check the expression of ARP7 and ARP19, and both decrease in MUR1 background. And very interestingly, when we provide the boron, ARP7 and ARP19 expression goes up to the wild like wild type. But why? It boron enhance the expression of ARP7 and ARP19. And we made a triple mutant to identify that the signaling cascade components, and we found that this in the triple mutant actually it's more severe comparing to double mutant, but where it's a very complex thing that in a triple mutant, here you can see in a triple mutant, when we provide boron, it does not rescue. But in a MUR1, as you can see MUR1 boron, it can rescue. So why in the triple mutant it does not rescue? It suggests that probably defect in pectin RG2 dimerization is mediated through ARP7, ARP19. But here it shows additive effect. So it's a very complex phenomena. It's a very difficult to answer at the moment how exactly it is going. So then next, because we know that ARP7, ARP19 expression goes down in MUR1 mutants, so we increase the expression of ARP7 in epidermal cells by using ATML1 promoter ARP7, actually this line, and it, as we expected, it rescued the defect. So whether this reduction in the expression of pins is due to the mirror image of reduction in ARP7, ARP19. So we checked it and we found that, yeah, it's a mirror image. So basically it reduced in ARP7, ARP19. So next I focus because it is known that oxygen and Brassenet steroid have a crosstalk. So I just show you the main component of this big Brassenet steroid signaling pathway. So in this signaling pathway, there are transcriptional factor BAS1 and BZR1. So when it is phosphorylated, it becomes inactive, and when it is non-phosphorylated, this is active pathway. So I check the expression of Brassenet steroid biosynthetic gene, and both goes down in MUR1 mutant. And when we provide the boron, it goes up. And because the expression goes down, then we provided the exogenous epibrasinolite, and it rescues the MUR1 defect. And BAS1D is a constitutive active brassinosterol signaling mutant, and it also rescue. It's very interesting that when we gave brassinosteroid inhibitor BRZ, brasinolite, it can, in the presence of BRZ, boron cannot rescue MUR1 hook defect. So with the help of Rusinova lab, we found that boron enhances PR signaling. So as I said that BAS1, when it is phosphorylated, so it's it's a phosphorylated by total BAS-P is readout of brassinosterate signaling. And when it is phosphorylated and ratio is more, then it is a weak signaling. And when the ratio of phosphorylated with total BAS1 is less than is a active strong signaling. So you can see that in the presence of boron, it increased. So boron enhances BR signaling. And with the help of Dr. Stephen C. Fry, we found out that brassinosteroids regulate RG2 dimerization. When you provide the brassinosteroid, it also enhances the RG2 dimerization. And in the last, how this change in the pectin RG2 dimerization is sensed. So it is known that Feronia, Arolas, and Thesis are cell wall integrity kinases, and Thesis is a negative regulator. So we made a double mutant, and very interestingly, it also rescues. So this means the change in the, and why it rescued, then we check the expression of ARP7 and ARP19, and it goes up. So this is the change. So this change in RG2 dimerization affect ARP7, ARP19, and it affects polar oxygen transfer machinery, and how polar oxygen and BR, it is a complex mechanism, how they regulate RG2 dimerization and differential cell elongation. And thank you everyone for the listening. Thank you.
Thank you. There are questions? Yes, Malcolm.
Okay, thank you. Yeah, very interesting talk. How do you think that the ARF7, ARF19 machinery is being, detecting cell wall changes?
Yeah, that's the main thing. Like